is the dura mater when we look at it from the outside, okay? I won't ask for arachnoid, but it would be this underside. When we take the dura off of the brain, we see that there are some thickenings in it. This thickening here runs the length of the brain, the cerebrum, runs the longitudinal fissure. This thickening right in here of the meninges is referred to as the falx cerebri, okay? This thickening here fits into the space between the occipital lobe and the cerebellum. Fits like that. This is called the tentorium cerebelli. So you can see that the meninges, in addition to creating a protective covering around the brain, also kind of segment the portions of the brain somewhat. We take a look at the external surface, and we see the cerebrum and the cerebellum and the brain stem, spinal cord back here, okay? When we look at the cerebrum, we see the frontal lobe. I can't tell you exactly where it ends, but I know that this is definitely frontal, so I'll stick a pin right up front. This area here is parietal. I can't tell you where it starts and ends. This area is the occipital, and this is the temporal. Okay, the insula we would have to dissect to find. That's the cerebellum, or cerebrum. The longitudinal, these don't probably match. The longitudinal fissure runs the length of it here, okay? If this were a human brain, we'd be able to identify then the lateral fissure or the lateral sulcus, okay? We keep looking. We have the pituitary gland with still some of its protective bony covering, the sella turcica, still present. Pituitary, this is one of the optic nerves here. There's the optic chiasm where the nerves cross over. Look for the hypothalamus above this or underneath this, okay? Here are the areas where the olfactory bulbs would be located. Yeah, this one has one, not much left, a little bit. No, actually it's gone, okay? So these will fit into the cribriform plates of your skull. And then they have axons, or actually they have neurons that pass down into the nasal cavity, okay? This is brainstem, spinal cord back here. We can see a bit of a bulge right here. And then we can see that this area is wider than this area here. Spinal cord, medulla oblongata. It's a little wider here. And then we see a little hump. That tells us pons, okay? So, spinal cord, medulla, pons, okay? And we're just seeing thickness changes. We would also perhaps see some histologic changes as well, okay? That means that we would see in the spinal cord, we're going to see that central gray matter and outer white. In the brainstem, we see nuclei of gray, and everything else is white. Okay, if that's probably TMI. Okay, getting in. Got to have a good specimen. All right. We look here and we see this white matter right in here. This is the corpus callosum. It also seems to come around like this, and then it sometimes looks a bit like a bird beak. Well, there aren't really good examples here. Sometimes looks a little bit like a bird's beak right in here. That is the corpus callosum, the connection between the two cerebral hemispheres. If we manipulate the corpus a little bit, we start to see a space. That is the lateral ventricle. And if you sort of loosen this and pull it out, that's the choroid plexus of the lateral ventricle. That is the CSF producer there in the lateral, okay? Let me have the bag. The third ventricle is not easy to see. It depends on the cut. There's the corpus. There's the lateral. The third is going to be a very narrow ventricle that surrounds this central circle. The central circle is called the intermediate mass of the thalamus, or the interthalamic adhesion. Pick one and stick with it, okay? And don't worry about the other one once you pick one. This is choroid plexus of the third. You can see some third in there now. There's third, comes around, third, there's choroid plexus in the third. Lateral's there, third is there. It is what it is. Okay, let's see what else we can see. I don't like these. Yeah, it's probably in the other bag, but yeah. I'm gonna go get that other bag.
I was just thinking that. I'm like, I'm so.
Actually touch it.
I'm gonna say go for it.
My hands will smell like sheep the rest of the day.
Might be
Don't want
a
that.
problem.
Yeah, if it's fresh, I would do it. Go for it. I eat it. Why not?
These have still some residual formalin in them, and then they have another preservative as well. So you don't want to eat these. Not that. I did have a woman from the Middle East who looked at these like, they look pretty good, because she ate lamb brains. Yes, this is a nice one.
Oh, there we go.
All right, the intermediate mass of the thalamus
They're so good.
is right there, and then we're seeing some choroid plexus in the third. Okay, intermediate mass and choroid plexus of the third. Okay, so now we're in the area of the diencephalon right in here. We know that the thalamus is diencephalon. There is the optic chiasm, so we expect to find the hypothalamus right in this area here. This is probably one of the bumps of the mammillary bodies right here, right there. And so we know we have hypothalamus there. We see the optic chiasm. We know the hypothalamus is in the general area. We line up the pituitary with, there it is there, with where it attaches. We know the hypothalamus is there, okay? This is the pineal gland of the epithalamus, okay? And then we see, as I probe, we see the cerebral aqueduct right there. And that is carrying CSF back to this fourth ventricle right between the cerebellum and the brainstem. Cerebral aqueduct, fourth ventricle. Sometimes we see a choroid plexus in there as well. This is the cerebellum. The cerebellum has numerous folia that are gray matter, nerve cell bodies, and then a central white matter known as the arbor vitae. Okay? There are other parts to it, but that's what we've got. You can see the cerebellum is wrinkled on its surface just like the cerebrum. The wrinkles on the cerebrum, the raised up wrinkle is called, pardon me, what's that called? Gyrus or gyree, and the indentations? Sulci or sulcus, okay? Let's see, so diencephalon. Once we start to see these two big bulges back here, the bigger one, the superior colliculus, the littler one, the inferior colliculus, then we know that those parts correspond with the midbrain. When we see the cerebral aqueduct, we know that that is part of the midbrain. So we had hypothalamus, thalamus, midbrain, pons, look for the bump, pons, medulla oblongata. Third ventricle, cerebral aqueduct, fourth. Cerebellum's folia, arbor vitae. What have we missed?
Yeah.
Okay,
Again.
now this is the pineal, okay? So if we sort of just manipulate the cerebellum a bit, we see two lumps
I see.
hidden in there. A big one and a littler one. Little one, big one. Little one, big one. Bigger one is the superior colliculus. The little one is the inferior colliculus.
Okay.
When you see the colliculi, you know you have the area of the midbrain.
What about the meninges?
The meninges, here they are. We tried to tear these, Aaron, so some of these are torn. But this is how it covers The olfactory bulbs stick out the front.
Okay.
That thickening separates the cerebrum from the cerebellum. This one has a very nice olfactory bulb, by the way. Okay?
The falx cerebri is this one?
Right, follows the longitudinal fissure, okay? You can see it has some blood in it. If I cut this, I'd actually be able to squeeze a little blood out of it, okay? It is the venous sinus of the brain. Brain is draining blood out of it. It goes into the sinus, okay? And then this thickening here is the separation between the cerebrum and the cerebellum, the tentorium cerebelli. Anything else?
Under the cerebellum, you list on this pink sheet the arbor vitae
Yeah.
and
Folia. Cortex, okay,
So
well.
is that the same as folia?
It is. It is. Just like in the cerebrum, the outer cortex, inner medulla, the cerebellum is the same way.
So is it FOLA?
It's folia, or if the pink sheet says cortex, don't write down folia. Just stay with cortex. Now, last thing. Cerebellum's here. We have the cerebrum, and it's been cut coronally, and you can see the inner white matter and then the outer gray. So the cerebellum isn't organized that much differently, okay? The basal nuclei you're not required to learn, but you can see then the lateral ventricle in its choroid plexus as I open it up. So we cut the brain like that, and then you can see the posterior horn of lateral ventricle. Yes? Right in there. Okay, I'll leave these out. If we want to talk about them some more, we can do that